Welcome to the course on biotechnology and molecular biology. My name is Pranav Doradla, and today we're going to be talking about plasmid anatomy and design. So you can see on this page a list of the topics we're going to talk about. Okay, so what's a plasmid? A plasmid is bacterial DNA that's separate from the main genome. So in a bacteria, just like in our mammalian cells, the bacteria has a chromosome, and we have several chromosomes, but the bacteria has one chromosome, and that's where all its DNA that facilitates all its processes and functions for survival is held. There's also plasmids, which are separate from that, which are often not essential for survival, but they help with certain stressful situations. So they can contain antibiotic resistance genes, which help bacteria fight off antibiotics. And because of that, that's how antibiotic resistance can develop. That's why that's a problem, because bacteria can exchange plasmids with each other. So let's say we create a new antibiotic drug that treats a certain bacterial infection. Well, if one bacteria mutates and it develops a gene on a plasmid that can fight off that antibiotic and render it useless, it can exchange that with other bacteria, and that can just spread throughout the population. So that's called conjugation. That's how they exchange plasmids. But we're not really going to talk about the infection here. We're going to talk about the biotechnology here. So what makes plasmids really useful is that they're small, they're easily editable, they can be easily placed into bacteria. That's called transformation, where you shock a bacterial cell with electricity or with heat, and that makes it porous. And then you can mix plasmids into the mixture, and those plasmids will go into the cell and then do their function in there. They'll get transcribed and then translated. The proteins will get translated and do whatever you want, produce whatever products you want. So that's why in many chemical reactors, they have yeast cells or bacterial cells or E. coli cells, for example, which have plasmids containing a gene for the protein of interest on there. And then these bacteria are tons and tons of bacteria in a vat producing the protein, and then that protein's extracted and used for whatever it's used for, whether it's a medicine or a protein for clothing or food or whatever. So the basic anatomy of the plasmid is what you can see on the screen over there. So there's a promoter for the main gene. The promoter is where the RNA transcriptase binds. Then there's an inserted gene in blue on the right-hand side over there. And on each side of it, it has a restriction site. So restriction enzymes are these enzymes that cut DNA, and they cut either straight or jagged with kind of like crossed ends. And so what you can do is cut the plasmid at each of those restriction sites so that it becomes a circle with a piece missing on one side. So it could be linear if it bends around. And then you take the gene of interest, the gene that you want to put into the plasmid, and cut it with the same restriction enzymes. Once you do that, then the jagged ends of the gene here and the one side of the plasmid here line up and base pair together. And then you can use ligase and some other enzymes to fuse the holes that are left over and insert the gene into the plasmid. So you can put whatever gene in there that you want. The only thing restricting you is really creativity. And there's some plasmids that have multiple cloning sites, MCS, and that's where they have several restriction sites in one place. So you can configure the size of the gene, you can configure the type of restriction enzymes that you're using. So you can put multiple inserted genes. You could have like 4 or 5 or 6 or even more genes in one plasmid that you want to transcribe. Then the origin of replication on the bottom is where the entire plasmid is replicated. And unlike our human chromosomes, which have multiple origins of replication, bacterial DNA, so the bacterial chromosome, as well as the bacterial plasmid, each only have one origin. Now, on the bottom left-hand side, there's an antibiotic resistance gene. So what researchers use this for is they'll put the antibiotic resistance gene in the plasmid, and then they'll transform bacteria with plasmids, and then they'll put those bacteria on a plate containing that antibiotic drug. So any bacteria that have not taken up the plasmid, that don't have the plasmid in them, that haven't been transformed, um, will not have that resistance gene, and thus they'll die. They'll get killed by the antibiotic. So this is a way to make sure that the only bacteria that you're harvesting are the ones that have the that are edited the way you want them to, that have the plasmid in them. Selectable marker is similar, but it functions kind of in the opposite way. Selectable markers don't kill the bacteria that don't have it, but they allow you to see the bacteria that do have the selectable markers. So an example of this could be like a fluorescent green protein, which makes the bacteria glow green. And then you can see, okay, on the plate, these green glowing bacteria are the bacteria that have taken up the plasmid. And that's just an additional way to tell which bacteria have taken up the plasmid. So because the bacteria are no use if they don't have the gene that you want them to have. So this is just an example of a system you can put in a plasmid to configure how much a gene is transcribed. So this is the lac operon. It has a repressor upstream of it, and you can see the promoter, and then 3 structural genes. So these structural genes code for different enzymes. And what happens is when the repressor is transcribed and translated, right, it's a protein, it binds to the operator right downstream of the promoter. So when RNA polymerase comes in and tries to transcribe the gene, this repressor physically blocks it from moving forward. And you can see that on the slide there. Now, when an inducer, like lactose or allolactose or IPTG, which is similar to allolactose, when that's present in the cell and binds to the repressor inactivates. So this induces the genetic circuit, so because it stops the repression, so it increases the transcription. And now the RNA polymerase is not blocked, so it can just proceed downstream and transcribe all the genes. So this is just one example, but there's several different ways to do this and create even more complex genetic circuits that allow you to even create logic, like Boolean logic, and have different things turning on and off at different times, depending on the concentrations of different things. So there's some work being done in cancer research, for example, where they're trying to use cell therapies to target and then kill the cancers. So these cell therapies are just normal cells, mammalian cells, human cells actually, and they have proteins on the outside of their membrane that can recognize cancer cells. And when they get to the cancer cells, they release certain proteins because they can also detect the concentration of certain chemicals that are released near the cancer cells. So when they detect the concentration of that using maybe a system similar to this, then the creation of toxic proteins is induced, and then that toxic protein accumulates near the cancer cells and kills them. So there's lots of really exciting work being done here, but this is just a very simple, low-level explanation of transcriptional switches and genetic circuits. I would recommend that you spend some time learning about it on your own time, because there's some really, really cool stuff going on here. Okay, bacterial versus mammalian expression. So you can put genes into bacteria, using plasmids, or you can put it in their genome. And you can also do it with mammalian cells, so human cells or yeast cells, or not yeast cells, that's eukaryotic, but human cells or other mammalian cells. And with bacterial cells, you have to remove the introns first. So if you're working with eukaryotic genes that have introns and exons, right? They can have alternative splicing. You have to remove the introns and only have the exons in one line, because bacterial cells don't do splicing, so they don't have the machinery to translate that. So if they try to transcribe and translate those proteins, it's just not going to work out properly. With mammalian cells, mammalian proteins work best in mammalian cells because, again, there's splicing and there's other more complicated folding, the chaperone proteins, which we talked about earlier. And it's just their native environment, so their functions are going to work better there. There's lots of other pros and cons, I'm sure. So that I'd also look up on your own time. Chromosomal versus episomal expression. Episomal means outside of, epi means outside of. So this is outside of the chromosome, outside of the main genome. So in bacteria, with chromosomal expression, you have to edit the genome with CRISPR or some other way of doing that. The stability is high and the copy number is low, because you can only put you only have one genome in which to put genes. Episomal, which would be plasmids, for example, in bacteria, you have to configure the plasmid, like we talked about earlier, and then electroporate or heat shock the cell, and then transform the cells with the plasmid. The stability is low, but the copy number is high, because you can have many plasmids in one cell. There's probably many more differences, so I'd also look this up. Okay, to recap, you can see everything that we learned about today. And next time, we're going to be talking about proteins and pathways and modifying them. So now we're really moving from the molecular biology to the bioengineering, to the protein engineering. We're going to learn about how to edit proteins, how to create new proteins that haven't been seen before. This is the really exciting engineering work that can change the world. So with that, happy learning, and I'll see you next time.